Well, thank you, Sonia, and good morning to everyone, or good afternoon or early morning for a few of us, depending on what time zone you're in. For those of you who do not know me, my name is Michelle Campbell, and I lead stakeholder engagement and clinical outcomes in the Office of Neuroscience. And so it is my pleasure today to welcome you to the 2022 FDA update. I have been a part of many of these update sessions, and it is actually one of my favorite sessions because every year it is something new. We are always talking about what your latest questions are, about advice from the agency, what is going on with our current thinking, with what we are seeing coming into medical product development. And it is really a truly hot topic or emerging concepts session that we put together to try to address the questions and needs of the entire COA community as we move forward. So it is my pleasure to introduce David Reisner, our Director of the Division of Clinical Outcome Assessments, to start this update. David?
Thank you, Michelle. Good morning. I'm David Reisner, Division Director, Division of Clinical Outcome Assessment, and FDA Liaison for the PRO Consortium. So it's my pleasure to kick off this first session of the PRO Consortium Annual Workshop alongside my colleagues who will be presenting FDA updates. Next slide, please. We have a great agenda planned for this morning. To start, I'll provide updates on the COA Qualification Program. Next, Robyn will discuss patient-focused drug development updates, followed by Laura Lee Johnson speaking about the CDER Office of Biostatistics, PFSS team, and a few COA-related guidances. Lastly, we will lightly cover a few topics of interest as an introduction to the panel discussion moderated by Michelle. Next slide. Before diving into the presentations, I'd now like to briefly introduce our panelists and speakers. Thank you. Robyn Bent, Director of the Patient-Focused Drug Development Program in the Center for Drug Evaluation and Research. Laura Lee Johnson, Division Director, Biometrics 3 in the Office of Translational Sciences, and also the OTS Liaison to the PFD program. And then lastly, Michelle Campbell, our Moderator, Senior Clinical Analyst for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience. Thank you, everyone. Next slide. We'll start off with the qualification program. I'll touch on the drug development tool guidance and talk a little bit about how the program operates, the 2021 metrics, and point you towards some program resources. Next slide. Next slide. In November of 2020, CDER published the final guidance describing the qualification process for DDTs, including COAs, biomarkers, and animal models, among others. The guidance describes the process under the 21st Century Cures Act. We also continue to add COA resources for requesters, including information on the qualification process, which can be found on our resource webpage. Both links, one to the guidance and one to our resource page, are provided in the presentation deck. Next slide. This is just an overview of the process. As many in this audience know, there's a three-stage qualification process for DDTs. The process is initiated with a letter of intent. The next stage is a qualification plan stage, typically including some qualitative results and a quantitative plan for review. And finally is the full qualification package, which is reviewed for qualification determination. Importantly, as indicated by the arrows underneath the formal milestones, in between these stages, we can also receive updates and meeting requests. So those interim submissions are important in order to maintain a dialogue as you continue to develop your DDT. An example might be a study protocol or revised SAP. Next slide. Thank you. In this table, I would just provide our targeted timeframes for each stage. We conduct a reviewability assessment for each milestone, which results in a letter issued to the requester with a reviewability determination. Our review begins with the acceptance of your submission and the issuance of the reviewable memo. Next slide. Now to the year 2021 and a few metrics that you might find of interest. We've been very active with qualification review at all stages in 2021, including holding pre-LOI meetings, reviewing LOI revisions, and restarting or withdrawing existing DDTs. We did receive fewer LOIs last year than previously, and although we worked with a number of requesters to refine their LOIs by, for instance, revising their concept of interest, none of those LOIs reached the acceptance stage by the end of the year. Next slide. The total number of active qualification projects is currently 64. And then when we look at those projects across therapeutic areas, we see a few review O&D review divisions who I would call our frequent flyers, but we also see a nice spread across a variety of the therapeutic areas. Next slide, please. Then when we look at the breakdown by COA type, we see that PROs dominate with a total of 41 projects, but all types are represented in the program. And our other category is an interesting and growing COA type that we are working actively on. Next slide. In terms of trends, we saw, as I mentioned, a decrease in the number of LOIs and also QPs submitted last year. However, we saw an increase in the number of interim submissions and meeting requests, such as the pre-LOI meetings that I mentioned. So the interactions with individual requesters, I would say, have gone up, but we look forward to new LOI submissions and potential acceptances in 2022. Next slide. Regarding resources, we awarded one DDT research grant and also deferred one grant into 2022. Last year, these grants support developers of DDTs that have an accepted letter of intent who are working towards the next qualification milestone submission. It's important to note for those in the audience for 2022, we currently have a funding opportunity announcement out, and the funding opportunity announcement number is on the slide for your reference, and further information is available on the FDA website. Next slide. For these and other updates, including newly qualified COAs, we encourage anybody in the audience who's interested to sign up for our COA QP email listserv. And the URL is on the slide for your reference. And then lastly, I think in my section is the next slide. The Division of Clinical Outcome Assessment is actively recruiting. You can expect to see postings on LinkedIn, on the ISPOR website, on the various FDA websites throughout the year as we solicit CVs, conduct interviews, and we onboard new reviewers. Ad hoc CVs can be sent directly to our mailbox, which is on the slide. It's decoa@fda.hhs.gov, and we appreciate your interest. Now we will transition to the patient-focused drug development section that will be presented by Robyn.
Great. Thanks so much, David. Thanks, everyone. It's great. Good morning. It's really great to be able to speak with you again. I'm excited to be here today to provide you with an update of some of our PFDD efforts. Over the next few minutes, I'm going to talk about patient-focused drug development. I'm going to provide you with an update on the patient-focused drug development guidance series. I'll talk about the standard core clinical outcome assessments and related endpoints grant program, and I'll wrap up with just a quick mention of the ICH patient-focused drug development reflection paper. So patient-focused drug development is really a systematic approach to help ensure that patients' experiences, perspectives, needs, and priorities are captured and meaningfully incorporated into drug development and evaluation. And one way that we're doing this is to facilitate and advance use of the systematic approaches to collect and utilize robust and meaningful patient and caregiver input to more consistently inform drug development and regulatory decision-making. And we're doing that partially through the development of these 4 methodologic guidance documents. These guidance documents describe how stakeholders, patients, researchers, medical product developers, and others can collect and submit patient experience data and other relevant information from patients and caregivers to be used for medical product development and regulatory decision-making. This first guidance, it's a joint guidance between the Center for Drugs and the Center for Biologics, was published in draft in 2018 and was finalized in June of 2020. It discusses sampling methods that can be used when planning a study to collect patient input. It also provides a general overview of the relationship between potential research questions and methods when deciding from whom to get input. This includes defining the target population and a development of a sampling strategy. Guidance 2 is also a CBER and CDER guidance. It was just finalized in February and discusses methods for eliciting information from individuals identified in Guidance 1. It presents a range of methods and established best research practices to identify what is important to patients with respect to burden of disease, burden of treatment, and the benefit and risks in the management of the patient's disease. In particular, the methods and best practices presented in the document can help elicit relevant information from patients and other stakeholders, such as how their disease affects their daily lives, what they find most troublesome, and the challenges, problems, and burdens of the treatment for the disease. Guidance 3 is a little bit late, but we're really working hard to get it out and hope that it will publish soon. This guidance is a collaboration between the Center for Drugs, the Center for Biologics, and the Center for Devices, and we hope that you'll find it worth the wait. It will address refining the list of concepts of interest important to patients for measurement. Given that not everything identified as important by patients, caregivers, and clinicians can be addressed by an investigational treatment or be measured, this guidance will address issues related to selecting what to measure for a medical product development program and identification or development of fit-for-purpose clinical outcome assessments to assess outcomes of importance to patients. We're working on internal and external training materials to go with this guidance that we plan to have ready shortly after this guidance publishes. And the fourth guidance in this series is also in progress. It will address topics related to incorporating COAs into endpoints for regulatory decision-making, including COA-related endpoint development, defining meaningful within patient core changes, and collection, analysis, interpretation, and submission of data. And so before I wrap up kind of talking about the guidance documents, I'd really like to make a little bit of an appeal about commenting on draft guidance documents, because I feel like this is an audience that will provide really good feedback. And I'm very much hoping that the PFDD guidance documents will be published for comments before next year's meeting. So to start, number one, we do want your feedback. It's a critical part of the process, but it can be hard when we get hundreds of pages of text, particularly if that text is in a PDF or something like that. So while we will review and of course incorporate feedback as appropriate, however the information is submitted to us, our ideal is to receive the feedback in a form kind of like the form that EMA uses for public consultations. And let me see, I'm going to try, let's see, to just put a link to that form in the chat in case it's useful to you. And then also number two, it's really helpful when you provide us with feedback about what you don't like or what you disagree with, and we definitely need that feedback. But if there's something that was covered well, please tell us that too. It's not that we need a lot of affirmation, but most of the comments that we receive are recommendations for change. So we don't necessarily know that something really resonates with a majority of people if we don't hear about it. And if we get a comment or two recommending a change or to remove that section, we may actually make that change. And so this may not be what everybody wants, and it can be hard for us to tell just reading the comments. Because we want to develop a document that helps us be clear about what FDA is looking for, but also meets the needs of stakeholders who are reading the document. So your feedback is important. Just kind of the more you let us know, the more we can kind of reach that goal. And so now I'm going to switch topics and talk about the Standard Core Clinical Outcome Assessment Grant Program. And so this grant program grew out of patient-focused drug development and things that we're hearing from our PFDD meetings. And the purpose of the Standard Core Clinical Outcome Assessment Grant Program is really threefold. It aims to help make incorporating patient perspective more sustainable. We want to enable the development of publicly available standard core sets of measures of disease burden and treatment burden for a given area. And it provides avenues to advance the use of patient input as an important part of drug development. And so now I'm going to talk a little bit about the grants that we have in the program. And it should be noted that as expected, different grantees have designed their grants differently. And so while we plan to have these core sets developed within the allotted timeframe, the grants were funded at different times, and all of the grantees are at different points in the development process. And so the first grant is the Migraine Clinical Outcome Assessment System, or MCOAS. And this team, led by RJ Worth and Richard Lipton, will develop and standardize a core set of endpoints and related COAs for use across migraine clinical trials. Next, we have the Clinical Outcome Assessments for Acute Pain Therapeutics in Infants and Young Children, COA APTIC grant, which is led by Kenesha Zimmerman and Bryce Reeve. It will identify COAs and endpoints for use when developing acute pain therapeutics in infants and young children, primarily those 0 to 2 years of age. We have the Northwestern University Clinical Outcome Assessment Team, or NUCOAT grant, led by Dave Sella. It will develop and validate COAs with applicability across a range of chronic conditions that assess physical function using patient-reported and performance outcomes. We also have our newer grants. Last year, we funded a grant titled Preparing a Clinical Outcome Assessment Set for Nephrotic Syndrome, or PREPARE-NS. This grant is led by Debbie Gibson and Devin Pipert, and the team plans to develop and establish a core set of COAs for nephrotic syndrome with a primary focus on fluid overload. And we have a grant titled Expanding the Observer Reported Communication Ability, or ORCA measure, led by Christy Ziegler, that will expand on the existing ORCA measure, a measurement tool created to assess caregiver observations of children's ability for expressive communication in nonverbal patients with Angelman syndrome who experience complex communication needs. They're hoping to expand the measure to cover 12 other neurodevelopmental disorders. One thing that really makes this grant program unique is the amount of FDA staff involvement. The FDA team meets monthly with the grantee teams to discuss the grant progress and next steps. We receive questions and documents from the grantee teams, and we ask questions. We don't always agree, but there are some good scientific discussions. On the FDA team, we have representatives from the clinical review divisions, the Division of Clinical Outcome Assessments, the Patient-Focused Statistical Support Team, and other statisticians who work directly with the clinical review divisions. Sometimes we have representatives from other medical product centers, and of course we have the staff from my office. It's really a collaborative discussion, and while I can't speak for everyone, I find the conversations we have internally and then with our grantee teams to be really interesting and very informative. And there are so many benefits to this program that I don't think we considered when we started. Several of our grants involve engagement from multiple therapeutic review divisions. For example, the ORCA grant, we have representatives from both the Division of Rare Diseases and Medical Genetics and the Division of Neurology 1. For the NUCOAT grant, we're working across divisions to develop a core set for physical function. The model conditions include conditions reviewed in the Division of General Endocrinology and the Division of Rheumatology and Transplant Medicine. The APTIC grant includes representatives from the Division of Pediatric and Maternal Health, the Division of Anesthesiology, Addiction Medicine, and Pain Medicine, and the Office of Pediatric Therapeutics. And this opportunity to develop these core sets that may work across a range of conditions is really very, very exciting for us. Another really interesting aspect about the grant program is the diversity of measures that we're seeing developed or adapted for use in the core sets. We're seeing PROs, performance outcome measures, and observer reported outcomes. Some of our grantees are looking at optimizing core sets for remote administration. And another interesting aspect of this grant is that we're seeing all kinds of different approaches being taken to develop these core sets. We believe that some of the grantees will be able to use existing measures as part of their core sets. Some grantees are looking at the possibility of modifying existing measures to use as part of the core set. And of course, there are some grantees who were unable to identify existing measures that included the concepts of interest identified by patients as meaningful and are therefore developing new measures. The process for each of these approaches varies, and we're still somewhat early in the development process, but we're very excited by what we're seeing, and we can't wait until the grantees are ready to make their milestone documents publicly available because they really are quite impressive. And so finally, I'd like to just, And so speaking of public availability, each of our grantees has a public website, which they're updating as the grants progress. This slide has some of the relevant links, but they're also available on the FDA Patient-Focused Drug Development website. And finally, I'd like to just briefly touch on the International Council for Harmonization, or ICH, Patient-Focused Drug Development Reflection Paper. The goal of this paper is to take steps to harmonize approaches, methods, and standards to advance incorporation of patient perspective in drug development globally. The goal is to build on existing work, not necessarily reinvent the wheel. And this reflection paper proposes to develop the development of ICH guidelines to address how to measure things that are meaningful to patients in a clinical trial, for example, through the use of clinical outcome assessments, and also methods for elicitation or collection of information on patient preference. The paper has been endorsed by the management committee, revised based on public comment, and is available on the ICH website. Because of the pandemic and the availability of subject matter experts, we've not begun working on the papers, but the work will move forward as resources allow. So thank you so much for your time. I hope that you have found my presentation informative. If you have any questions, please let me know. I'm now happy to turn the microphone over to Dr. Laura Lee Johnson. Laura Lee.
Hi, everybody, and nice to see you again. So I'm going to tell you a little bit about our patient-focused statistical support group, also known as PFSS, and go through a few guidances of interest, including one hot off the press. So these are the pictures up at the top of our folks on the patient-focused statistical support team. Lily Gerard is our team leader. You'll be hearing from her later today. Also, we have Monica Morel, Weimong Wang, Marian Strazeri, and Xing Wan. Their team home is inside of our Office of Biostatistics in CDER. However, the scope of who they work with is much broader. They actually do consultations across all of FDA, every single center. And their primary work, though, is within CDER's Office of New Drugs, or O&D. They work very closely with the Division of Clinical Outcome Assessments. Now, we both groups have psychometricians, but you may wonder, when would DCOA perhaps consult PFSS? So they typically will get requests for help from our clinical groups, from the statistical teams, or from DCOA. So if DCOA needs, for example, a secondary review on psychometrics and meaningful change, that's when they would go to PFSS. So we do work a lot together. Lily and David are talking very regularly, so you may see a lot of people or a few people, but just to give you an idea. So what do? They provide statistical and psychometrics input on the development and use of COAs, and also for other patient experience data across all therapeutic areas and submissions. So this can be everything from proposals and contracts. It could be grants, such as the ones that Captain Bent just mentioned, also drug development tools and the Qualification Program, and then NDAs, BLAs, and INDs and their equivalents in the other centers. So it's across a large range of therapeutic areas and different types of submissions. They support a matrix of statistical reviewers and team leaders that we have inside of CDER's Office of Biostatistics, and people in that matrix receive additional COA, digital health technology, and patient preference training. So we are there to help back them up. So you may not see just the people from PFSS, you might see people from the Office of Biostatistics that are part of this matrix that will be providing additional support to their statistical reviewers and their teams. This group also leads and contributes to policy and guidance development, outreach related to COAs, and patient experience data. So they're very involved in the patient-focused drug development efforts. They're also involved in the therapeutic area guidances. So these folks are part of also a lot of research projects to advance methodology for PFDD tool development and to facilitate regulatory decision-making. They provide and facilitate internal and external trainings and workshops on PFDD-related statistical issues. So they've got a lot of communications parts as well. So we are hiring, as you've heard both from David, and I know all of our parts of FDA are also hiring, but if you're interested and you're looking for a new experience, we're looking for COA and quantitative patient preference expertise at all levels. So we are interested in hiring and have several openings from reviewers through supervisors. And so please contact us. We have a general mailbox inside of our Office of Translational Sciences, and you can also CC me, and we can help make sure that everything goes where it needs to go. Now, I wanted to transition now to a handful of guidances. Many of these, so on this first slide, I'm going to go through the guidances related to COVID. So we have a link, and I'll drop them into the chat when I'm done here, that goes to every single FDA COVID-related guidance. From there, you can search for anything you want. This middle one talks about the conduct of clinical trials for medical products during the public health emergency. And we've talked about this the last few years. It's being constantly updated. And many folks and groups are actually referring to this guidance when they're trying to handle and think about how to deal with trials and data from trial sites in Ukraine and the surrounding areas. Now, next to this is also the Statistical Considerations Guidance, which discusses, based on our views in 2020, how to approach the analysis of trials where study sites were closed or patients were not able to make some of the study visits, or that data collection methods changed for some patients. Now that some of these trials have matured from earlier in the pandemic and have been coming into the agency for review, we're also not seeing that the pandemic is really a blip, but now we have a longer standing issue. Thoughts on how to handle the intercurrent events is evolving, and it's important to speak to the statistical staff at FDA before unblinding data to make sure that the statistical analysis plan is up to date, not only with the sponsors, but also the agency's current thinking. And many sponsors have done a lot of interesting work in this area, and we're very thankful for it. Having frequent and recurring data audits has also, it's always been important, but it's very important now, especially we're seeing this as an issue with the war and sites that may not exist anymore for companies to go and audit the data. So again, talk to your regulators about the specifics of your situation. I think that's going to be very important as we move forward. So 4 guidances of interest here, and one that just published today, I think. First, we have 2 CDRH and CBER final guidances. The first is principles for selecting, developing, modifying, and adapting PRO instruments for use in medical device evaluation. Now, CBER also handles some medical devices, so you will see that they join together on many guidances. They also finalize their guidance on patient engagement. These are guidances that we, even though CDER, the drugs, are not on these guidances, we've worked very closely with the personnel and our sister centers in order to make sure that all of our work is interlaced and that things are in alignment with our PFDD guidance series as well. This Oncology Center of Excellence released their core patient-reported outcomes in cancer clinical trials draft guidance, and we also have 3 center guidance on digital health technologies that is out in draft, and you'll be hearing more about that during this workshop as well. Now, a 5th guidance published this morning on diversity plans to improve enrollment of participants from underrepresented racial and ethnic subgroups in clinical trials. And this recommends that sponsors of medical products develop and submit a race and ethnicity diversity plan to the agency early in clinical development based on a framework outlined in the guidance. And this builds on final guidance that was published in 2020 on enhancing the diversity of clinical trial populations. This guidance applies to all medical products. And if people want, we can add a link to that into the chat later as well. So, topics of interest, transitioning a bit here. I'm going to talk a little bit about percent change. Many times we think about percent change from baseline, but not always the place where we start. So one thing that you will repeatedly hear from the FDA is that you need to give us evidence from the context of use as to how you're going to define meaningful change. Now, some groups will come up, they'll say, well, 15% change from baseline is a meaningful change, 30% change from baseline, 50% change. And they keep upping the number sometimes, but not thinking back to what's actually clinically meaningful for the patient. Now, I'll put a little pause here and say, 25%, 100% resolved. So if everything is cleared, frequently that is clinically meaningful. If everybody has become completely clear or well, then you probably don't need an anchor. We can probably accept that. But all situations vary. But I think it's really important to understand, how are you determining what is clinically meaningful to people? So I'm a mathematician by training. We're going to do a little check. Let's say 0, 5, 10. Somebody starts at 5. They move to 10. And this is a good change. It is an improvement. And it's 100% change, but they've moved 5 points on your scale. Now they decrease. Somebody started at 10. They moved to 5. This is not a good thing. They've lost 5 points. 50% change, negative 50%. So now we're going to have a poll. You are the FDA reviewer. Now, I know you've got a whole study like this. FDA doesn't see many single-arm studies, but I'll also say in RCT, it's not that uncommon that when you see one thing in the absolute change and another in percent change, so you've got to make a choice. So T, can you put up our poll, or do I need to give you back? Here we go. All right, everybody, please vote. You got data that looks like this: no overall change or an overall improvement. Have to choose one. See, how are we on the time? Can we close it? Okay, 69% of people said no overall change. 31% said an overall improvement. So here's our problem. In both cases, the absolute change is 5. And yeah, the average change of that original scale is 0, and it indicates that there is no overall change. The percent change is very different, though. There, on average, you got a 25% overall improvement. And this becomes the problem that we have, right? So at least there was no, there are no zeros at baseline. And then if the patient started at 1 or 2, is that going to be meaningful? If you have a 15% change, a 30% change, a 50% change, what's actually meaningful? What if the scale was 0 to 1,000 or 0 to 100 and you're looking at those 5-point changes? Is that meaningful? And so this is what we have to grapple with. Now, some of the statisticians will sometimes come to you and they'll say, hey, listen, I can get rid of your 0 problem at least. Put the whole range, that biggest number, in the denominator. We don't see it that often, and it actually addresses a different idea than percent change from baseline. Now, other people will say this is a multiplicative rather than an additive treatment effect. Usually we don't know what it really is, but even so, there's lots of ways that you can handle that, including with logarithmic or other types of transformations. But I'll tell you another thing to watch out for is that randomized withdrawal designs that say you can go back on therapy. So these are the ones like you get randomized up front. Now you have supposedly hit a response, and they're going to withdraw the product and see if you get worse. And then once you get a certain amount worse, they're going to put you back on the product. But when you define that certain amount worse by percent change, you can sometimes see patients that are so sick at this point, so symptomatic, they would have qualified to be back on the study under the original enrollment criteria, but they're not allowed to be back on therapy. So if you want to use percent change, you still need to also keep in mind that a lot of these scores are going to be non-normal distributions. They're really challenging to model compared to your landmark scores or change from baseline. So in summary, big font for a reason. If the analysis is going to be using percent change, you need to think about all the numeric scenarios and what they mean across the entire spectrum of people that are going to be providing data. And you need to provide to FDA evidence and the rationale for using percent change. And don't point to, somebody else did it or they said you could do it. But you need to actually provide why this is going to be clinically meaningful, and is the method of determining meaningful change directly considering the patient voice. So a brief minute on pure CTCA versus CTCAE. Grade 1 diarrhea, grade 2 diarrhea, that's CTCAE. People have come to us and they keep asking, even though we have a 2018 publication, they keep asking, are we expecting these to travel together? And the answer is no, we do not expect that CTCAE will travel with PRO CTCAE. And that includes our clinical site inspectors are not going to be expecting that either. So the watery stool is a question. Think about this diarrhea question here. Like almost constantly, I could come into a trial already almost constantly having diarrhea. And I maintain that throughout the trial. The CTCAE is asking very different questions. And so that's something that we recognize and our inspectors recognize. So I'm going to turn this over now back to David.
Thank you, Laura Lee. I'm now going to cover a few topics of interest that people seem to have questions about, and maybe I can provide a little guidance on lessons learned or things to consider. Next slide, please. In terms of anchor-based analyses, anchor-based methods are the primary methods we use to interpret meaningful within patient change. The anchor-based methods should be supplemented with ECDF and PDF curves, very helpful. The graphical summary in terms of interpreting responder thresholds across the entire range. But other methods, for example, exit interviews, can also be explored when anchor-based methods are challenging to interpret. Although I would point out that exit interviews are becoming more common across therapeutic areas and outside of rare disease, but exit interviews and surveys are not a substitute for careful planning, which may include deploying multiple anchors prospectively in your program. Next slide. One important aspect of anchoring is that an anchor scale should measure the same concept as the target efficacy endpoint. For instance, the aspect of the disease that's targeted in your endpoint. That provides the most direct evidence for interpretation. Next slide. Also, in terms of interpretation, an anchor scale that uses a 0 to 10 numeric rating scale is not directly interpretable, whereas a common verbal rating scale, such as none, mild, moderate, or severe, is easy to interpret in most contexts. Next slide. The anchor's recall period should be consistent with the assessment time period of the target efficacy endpoint. And by that, I mean, it's helpful that the schedule assessment reflects common schedule. The anchor may be scheduled to be collected at the same time, but the recall period should really be geared towards the endpoint and how you're going to be interpreting that. So it may be that the recall period of your anchor is longer and covers the entire assessment period in which you're using the assessment to construct an endpoint. So if you're looking at a weekly average, a recall period of 7 days for your anchor seems to be appropriate. Next slide. A few things about interpreting clinically meaningful patient change. One, anchors should not be double-barreled. And when you construct parentheticals, which may sometimes be very useful. You should not invoke other aspects of the concept. For example, if your parenthetical for a severity anchor mentions frequency or impact or interference, there's a contradiction between the concept you're trying to test with your targeted efficacy endpoint and then the anchor you're using to interpret that endpoint. For ease of interpretation, another point which is helpful, I think, is that the ECDF should be consistent after standardization and reverse scoring. We sometimes see, perhaps programmatically, the CDFs are generated in a consistent manner, but in fact, the direction of interpretation has changed post-standardization. Next slide. Another topic of interest is alternative scoring. Alternative scoring provides an approach to better understand problematic items or structure. Alternative scoring is different than an instrument modification, though, because all the items should be presented, understood, and completed without impacting the developer's instrument. Next slide. Supplemental analysis with the alternative scoring may be helpful in understanding, for example, poorly performing items that might exhibit ceiling or floor effects. However, consider whether those items are important throughout the treatment period in assessing improvement or worsening. And then lastly, when you're employing an alternative endpoint in an endpoint model as versus supplemental analysis, there's necessarily a higher burden in terms of the evidence you would bring to demonstrate that the endpoint is measuring what you say it's measuring. Thank you. I think that ends the topics of interest section. And I turn it over to Michelle to moderate the panel discussion.
Well, thank you, David, Robyn, and Laura Lee for presenting and providing these great updates that I know our attendees have been already messaging in some questions. So I want to start off with, to our attendees, if you have a question, please raise your hand, and then T will, I will call you, and T will unmute you so you can ask your question. I do ask that you please tell us your name and what organization you are part of with your question, and we will direct that. I also want to say that we've got a lot of questions about will the meeting slides and recordings be available? Yes, they will. They'll be posted on the Bureau Consortium website sometime in May, both the slides and the recordings. And so that was a common question we were seeing during the last session. So I'm going to start first with the first person. We've got a couple hands. We've got about 25 minutes or so to ask questions. So I'm going to first call on, and I'm sorry, I apologize now for any mispronunciations. Elva Cha.
Hi, everyone. Thank you so much for this very informative set of presentations. My name's Elva Cha, and I'm with Chiesi, USA. We work a lot in the rare disease space or ultra rare space. And with regards to clinically meaningful thresholds, where there is none in the disease area, how does FDA view using a rule of thumb like half of the standard deviation at baseline?
Hi, this is Laura Lee. And thank you for your question. So we, in our guidance, say that that cannot be how we would define meaningful change. So that can be used as a supportive group, but we're actually going to have a whole session this and another right after this talking about meaningful change. So I'm going to defer most of that to their session.
Thank you.
Thank you for your question, and hopefully our folks who are going to be presenting in our panelists in that next section will remember what was just asked and see if they can try to answer that as well. And next, I would like to ask Hillary Brines to ask her question.
Hi, this is Hillary Burns. I'm at ICON Clinical Research. I have two questions briefly. One is, how do you define a dossier? It seems to have changed in recent advice. And then the second question is, if there is, if there's a request for, will a request for qualitative evidence with patients to support content validity in novel conditions related to a condition for which a COA measure has already been established, might it reasonably be waived? I hope that was clear.
So I'm going to try to make repeat what you just said for my colleagues, make sure that we got that. So you first were asking about what are the dossier requirements that you seem to sense that there is changing in, I don't want to use requirements, but in air quotes, what needs to be included in a dossier for review to support a COA that's going to be supporting a clinical trial endpoint was your first question. I believe your second question was more specifically about the use of qualitative evidence in a condition where there's no established qualitative work, but the COA has been used in other diseases, and would that need for that qualitative work be waived? Is that correct? And I think we probably want the clarification more on your latter part of your question than the first part. I think we understood the dossier question.
Yeah, I think you captured it well.
Okay. David, do you want to start? And then I probably will also turn that back over to Laura Lee as well with some thoughts. But David, do you want to start? I guess we'll start with the first question about the dossier and what should be included in a dossier review. We'll start with that question first.
And now, it was specific to clinically meaningful change or more broadly?
More broadly, just how do you define a dossier? And it seems to have changed some in recent advice to include partial evidence about a measure.
Yeah, I mean, I would point back to the original PRO guidance where there was good direction on the ECT structure, and also more recent developments related to electronic document room. It's probably more detail we can go into the panel session. And in terms of the second question, which I think sort of addresses generalizability of qualitative research into new indications and populations, it in part depends, I think, on the historical data that are available. Typically, we would ask for some de novo qualitative research in your specific context of use, informed by the patients you're studying. I mean, one of the really exciting things about the patient-focused drug development grant program for standardized core COAs is that the approach incorporates the idea that studying a set of related indications can inform those indications, but also adjacent patient populations. So it's a principle that's important. But there may be items that don't settle well with your particular patient group, and you might want to know how those items perform. Consider alternative scoring, either as a supplemental analysis or in some circumstances, an alternative endpoint. So I'm not sure that we would assume that, this is a broad ask, right? That all possible concepts of interest in all patient populations would generalize. And I think to de-risk your program, I think you want to do that check. Confirmatory work with patients, qualitative research wouldn't necessarily be of the same scope as a de novo instrument that's never been studied with the patient voice. I don't know, Laura Lee, if you wanted to add something.
I think that covers it fairly well, and you'll probably hear additional feedback on this throughout the meeting as well.
Yeah, thank you, David and Laura Lee, and thank you for your question, Hillary. I think David made a great point that the PRO guidance is still the current guidance, and in the appendix, they actually provide the outline of consideration for what should be included in a dossier, and those sections are still relevant and important. And also in terms of, going into coming from a division perspective, when we do see new diseases and COAs that are coming from different diseases into a disease or disorder that's never been used before, we do want to make sure that is actually capturing things that are important to patients. So that evidence is still going to be very important for us to understand that why is the selection of this COA, as David said, the context of use is critical. So we do want to kind of see some of that early evidence to understand why a COA is being used to support an endpoint, and with that COA having not been used in that disease prior. So great question, and thank you for that question, Hillary. I am now going to ask folk if they would like to ask their question.
Yes, thank you very much, Michelle. I'm Folke from AstraZeneca, statistics and patient report outcomes. So I have so many questions, and it's been great so far, but I'm going to focus on the question that kind of relates to this part of the session. So we're very interested in understanding how the agency views looking at distributional differences instead of change, even when dealing with COAs where historically and traditionally, change has been the main topic of interest. And this is mainly, of course, for statistical reasons, where you avoid some issues with ceiling effects and floor effects, but more generally, if that's the main analysis, does that pose additional issues?
Laura Lee, I feel like this is a question right for you, up your alley. So, and it's a great question. I think as we're trying to find answers in particularly in our rare diseases of trying to interpret things. So Laura Lee.
Sure, and so I am trying to make sure I'm interpreting the question correctly. So there are a lot of different types of statistical analyses that can be done. And I want to distinguish this from one of the questions that the first questioner asked, I think it was Elva, about trying to use a distribution method to actually define meaningful change. There are other types of statistical analysis models. So like, if you think about ANCOVA or other things, those types of methods are different. And so the distribution-based methods, when you think about like effect size or proportions of standard deviations, those can't be primary evidence for identifying the range of meaningful change thresholds. But if what you're talking about instead is trying to use an entire data analysis method that's comparing distributions, that's fundamental for a lot of the statistical analyses that we see. So it's a fine distinction. I'm probably off the top of my head, not making it as clearly as I'd like to, but it is something that will hopefully be clearly made be made clear in our guidances that are coming out, because there are many types of analyses, especially the non-parametric analyses, that really are comparing data distributions and trying to look at shifts in the data. And if that's what you're referring to, I think those are, depending on the actual estimand that you're trying to address, that those are valid statistical methods that we see used. But that's a different type of question.
Right. You actually got.
Did I understand the question correctly?
I mean, you actually got to the first, to the other one of the other questions I want to ask, but sticking to this one, yes, I was not talking about distribution-based methods to derive magnitudes of meaningfulness, but rather comparing groups. So comparing, for example, treatment groups in terms of their distributions at a given time point instead of their path to get to that time point. Relying solely on, we have randomization, we have a large study, given everything else being equal, they should be at the same distributional level at baseline. Then even if we don't look at change, we're implicitly looking at change by comparing their distributions at, say, 6 months or a year later. Yeah. I was asking if that's an acceptable approach as a main analysis. I know it's acceptable, it's supportive, but as a main analysis.
And so again, depending on the exact situation, I don't want you to say, but she said this, without knowing the entire details of the question for your specific trial. But yes, we do see analyses like that for trials as primary analyses, and they can be considered. In fact, sometimes we recommend them to be the primary analysis.
Thank you very much.
So that's a landmark.
Well, thank you for your question. And before I go to the next question, I was also reminded back to Hillary's question on examples of outlines for the dossier that the full qualification package outline that is located on the COA qualification website, well, it is for qualification. That outline also does list the evidence that is needed for that submission, and it is extremely similar to what would be needed in a dossier. So another resource to kind of think about what is that information and evidence that the agency would like to see when submitting work to support the use of COA to support an endpoint. So just something I want to add there as another option. The next person who would like to ask a question is Ann Berg. So Ann, what is your question today?
Hi, my name is Ann Berg. I'm at Northwestern and Combined Brain. And thank you for this fantastic meeting. The question is for David Reisner. Early on, you showed the number of LOIs that have been coming in and been accepted. And I was wondering, what leads an LOI to be particularly successful and go forward versus not?
Okay, thanks for the question. You know, it does vary, and the work that we do to refine the LOIs, improve them, so we can present them for acceptance through our internal process. A lot of times it focuses around clarity in terms of the upfront work, the important aspect of health, the context of use, the concept of interest, right? So it may seem obvious sometimes with a COA, but I noticed with DHTs in particular, the upfront planning is important. Because it's hard for us to really review and evaluate the high-level proposal that is put forward in an LOI without sort of knowing what it is you want to do. And I would really think that's probably the best advice in terms of planning a successful LOI. And I mentioned PO-LOI meetings earlier. And so within the COA qualification program, if you've made a good faith effort, looked at the template, watched the videos on our website, and you have open questions, scientific questions you'd like to talk about prior to submission, then you should reach out to our mailbox to see if we can arrange a meeting.
Okay, thank you.
Yes, thank you for your question, Ann. I think I would like to add that one of those critical things that David said is really that context of use and your concept of interest is extremely critical. And we need to remember that our COAs have to be tied to an endpoint in the end. And so that needs to be kind of thought about. We understand that the context of use may shift over time as the data emerges, and that's okay. But I think from experience where we see programs that are able to move forward is if they have a good understanding of the disease, what they want to, the COA they want to use, how it will add to the science, and really what is it going to do, and is it really clinically meaningful as well. So those are some other things, but I really want to emphasize David's point on the context of use and the concept of interest. Those are the first two things that are always looked at when the letter of intent comes in. And that's also, important for a division when we are brought in to say, does this make sense for your disease that you guys regulate products for? We also very much want to know that makes sense as well. So thank you, Ann, for that question. I'm now going to ask Lindsay. You have a question?
Hi, I'm Lindsay. I'm a fellow at Novartis, and I have a question about removing DDTs that was briefly brought up during the DDT process, and just what that looks like, and if it's because they're being replaced, and what considerations you take into account.
So Michelle, I guess that would be a question to me. The question was regarding DDTs that are progressing or have been withdrawn by the questioner. I think was the question. Some DDTs have been in the program a while, and I think that as we've talked with requesters about the information that we're looking for, working through either the acceptance of a milestone submission or the actual review. Requesters have the option to withdraw that DDT if they don't see that the original need is there, or in some cases, the requester group that began the process is not available any longer. So there's a lack of scientific talent and other resources to move the DDT forward. We do accept DDTs from all sorts of stakeholders. So not only industry, but academia and patient groups and small companies. So the capacity to continue to move the DDT forward probably varies. So some requesters might choose to withdraw or choose other avenues like a newly formed consortia as an avenue to move their DDT forward.
Okay, thank you. And then I just had one other question about when you're looking at psychometric evaluations, about what percent would you consider to be a floor or a ceiling effect? Would it be like more than 20%, or how do you decide that?
If that's a question to me as a follow-up to my slide, I can comment briefly, but I'd also like to turn the question over to Laura as well, because the PFSS team is heavily involved in how we look at the evidence quantitatively. I don't know that it's difficult to work with rules of thumb. We see a lot of them. We see them in terms of reliability or treatment effects, and they are just general rules. I think it's probably, I should be more cautious and say it really depends on what you're trying to accomplish. When we include in context of use the design of the trial, the way you characterize the patient population, whether it's enriched or not, the caution that I put into that slide was that we want to look both at the item and the instrument level. And so if the instrument's performing well, and you have some items that have apparent floor and ceiling effects, you should really step back and think about whether you're going to need those in order to be sensitive to change, whether it's worsening or improvement. And so we will look at the item level analysis. We're very interested. But I think before you say propose an alternative version of an instrument, you should step back and think about the implications of any mods or say an item reduction you were considering.
Yeah, and I agree with David. I don't think, there's a strict rule of thumb, but the other thing that I remind folks of is that you have to think about the scores as a whole. And so when folks are throwing out items because they're not, they have a ceiling or a floor by item, it can become a problem because that could actually be what's really differentiating across that entire spectrum of severity. So that's another way to put some of that information. So be careful about what you're actually trying to make those decisions on, that's a ceiling or floor. And I'll leave it at that because I know we're running out of time here.
Thank you. Thank you for your question, Lindsay, and thank you, David and Laura Lee. So we have probably time for just one more question, so we can wrap up on time and make sure we can give you a full break. So I'm going to ask Christina O'Donnell if she would like to ask her question. And I do know that we have more people who have questions, and we have more people who are posting questions in the chat. So in my wrap up, I'll try to speak to a couple of them if I can. But Christina?
Christina, you're muted.
I'm so sorry. Can you hear me?
Yes, we can.
Sorry, I'm sorry, I was double muted. Thank you so much. From ICON, and I was hoping you could expand a bit on the mention of exit interviews. So how can this kind of qualitative data support meaningful change findings from a trial? What kinds of questions should patients be asked? And would this differ based on whether or not there's a corresponding screening interview?
I'm sorry, what was the last part? A corresponding
screening interview.
Yeah.
So I think some of this is actually going to be discussed in the next session, if I remember correctly. So I don't know if we want to hold on exit interviews, but maybe I'm wrong. Yes, I think we are going to be talking about this in the next session. Somebody's messaging me. Oh, screening interviews. I don't know, David, so is your question, is it only exit interviews, or can you do interviews prior to the clinical trial?
Oh, yeah, and I think exit views, and sorry, I didn't understand the full context of the question. Exit views in part are a sample of convenience, particularly in rare disease, patients are so precious. If you've touched them in terms of the conduct of your trial and you propose additional procedure like an exit interview to them, that seems to be very efficient for sponsors, and we see those proposals. But you can run a parallel standalone qualitative research study anytime. Those are fine, obviously. And there may be, just to note, there may be patients that don't fully qualify for your study, but are otherwise a good match for your context of use. And before they leave the site, you might want to talk to them about participating in your COA development research, because again, they're precious. So there you go.
Well, I want to thank everyone for their questions today. I want to apologize to those who are still raising their hands. We unfortunately do not have really time to take any more questions. There's a whole bunch of questions we've got chatted in. I do think that some of the questions that people want to ask will be covered in our other sessions of today and tomorrow. And so hopefully you'll get some of those questions answered from other sessions, and we will have FDA panelists on those sessions as well. I want to, there was a couple questions quickly about resources, references for alternative scoring. So I'm sure we can maybe pull some of the questions that were there in terms of resources and links and things of that nature. We can definitely pull back and perhaps provide to the PRO Consortium for when they post their information. This has been a really great conversation. This, like I say, the session is different every year. There were some really great questions that we will take back, the questions you have to help inform us.